tremendous honor, and especially thank Bengt, because he was the inviter, and I've always been fascinated by him, and so I sometimes don't make it to the meetings, then we get mad at each other, and so I couldn't possibly not get here, Bengt, and then Philip Ball is there next to Bengt, and he's one of my heroes in terms of natural world. What is it, and why, and how can we even understand it? So he's got the inside track on a lot of things, starting with water. In case you had any doubts, there isn't anything like water. I think that's more important to life, but also to all kinds of things. And so that's a story in its own right. Okay, now how do I? Okay, I'm going to try. What are you showing there? Oh, this is the slide for click chemistry with. Oh, let's see it down there. Okay. So I just felt like Hartmut Kolb and M.G. Finn, they were colleagues. My one postdoc with Kolb and long-term postdoc, and later working in a company we started in M.G. for graduate student years even. But he came, Lerner loved him, and so he hired him at Scripps. So we were a happy family out there. And we wrote the latest Nobel thing, which is about 2 years late, but it's good because MG came back and Hartmut came back to help me write it. I can get a lot of details, but I can't make it really, really powerful the way they can, and the way Philip Ball can. But and Bengt is a very creative physical chemist, and he doesn't mess around. He tries to get really tough problems, and he maced me once. It looked like a Nobel Prize to me, but of course it's hard for them to win a Nobel Prize, I think. But once in a blue moon, something happened. Samuelson was one, right? Yeah. Okay. Well, let me try to get on with the excitement of, because if you don't know about The Three Amigos, that's the movie with the Carisi comedians, and it's a fabulous movie, even for adults. But, okay, this is not something I put there. I didn't really put it there. Okay, that's for later on. I couldn't get it into this. I lost about 6 lectures today trying to get them finalized, and they keep dropping stuff. So let's try and see what we can find here. This is a typical nude mouse display of tumors growing on its back. And that's what, even it's written into the patents and into the government's what's useful to determine whether cancer can be killed or not. And I think none of the people anymore really respect that. It's too much of an organ change from the 2 species. But anyway, here's the thing. It grows a human cancer tumor. This is the matrix, or the vehicle, up here. Well, okay. Up here is the, can I go back? No, I can't. I could just use this, can I?
Yes.
Okay. Yeah, good. Thank you. I'll get this sorted out, but probably waste a lot of time. And I'll just stop when I have to. Okay. What you can see, there's one funny one here, and it was done by the people who did this work and it was there. And I looked at that, and I went in with a magnifying glass on the computer, and it's nothing there. I've never seen one of these things. Like, you see a lot of these in the world if you're a natural scientist, because everybody's working on cancer, and this is one of their favorite screens. So that one, and that turned out to be true for this compound that Jaja and Min Luit had been making hundreds, or actually 400,000 of, I think. They made a lot of compounds by the QAC reaction. And that's one of the things that I come to realize in this exercise of the last 6 months, is that the click reaction philosophy, what it led me to, is something which I try to share with you today, but I haven't managed to get the slides in perfect order. So I may send you some article that's written that does it better. But right now I can tell you, the one thing that I learned from the click chemistry that I couldn't have learned any other way is what a perfect reaction is like. because as good as reactions can be, and you know how much pride you feel when you get a really good yield, you get even 99% yield. Well, I don't have those slides lined up for you today, but if 99% after 5 steps, you're already hurting for a mixture, you have a mixture and you're having not great results. But if you have 99.995 or 999, you can go 1,000 steps and still have 99% yield. That's something which I didn't quite grok properly. And so what that means, and it makes sense, because if you're at high 9, it's 0.99999, and you multiply that times itself, that's 2 steps, 3 steps. Pretty soon, you realize nothing's coming off with that kind of multiplication happening to itself. It's just not moving. So that was a sign to me that there could be perfect reactions, and that we were always just reaching for great quantitative yield, this or that. But we figured out that the 2 we found by accident, QAC and suffix, are both perfect reactions. I can't guarantee any other reaction is as perfect as them. So that's weird, right? But I'm telling you, we found them both within 12 years of each other, and we found them because the QAC reaction itself was perfect, and it could go 1,000 times and still be 98%, 95%. So that's polymer chemistry. But I'm sorry, I don't know if the slides are going to satisfy me too much, but I'm going to try. Okay, this is my muse. What's what made me love chemistry? Because a local habitation in a name here, a periodic table. And I got really in love with it in grammar school, I guess, but so I memorized it. Big deal. It's easy to do, but I always felt uncomfortable if I didn't have a little plastic one in my pocket, in my wallet. You know, I get really sort of break out into sweat, you know, and sorry about that. Okay, so here are elements that I fell in love with, and copper, of course, just intruded itself so big into my life, but I also love a lot of these metals that I didn't circle. But the main group is so important to life on Earth, right? The main group is really where the heavy lifters are for life. But it's all P-block group, a P-block group. And we draw, when we do carbon chemistry, what did I do? When we do carbon chemistry, we're used to having perfect reactions that are damn good. None of them are perfect, though, in my opinion. And the Diels-Alder, the Aldo, or the Michael reaction, they're very good, but they're not perfect. And the reason why they're very good is because if you have pi bonds, you're exposing yourself to the world. I'm an atom in here, a couple of atoms. If I have pi bonds, they're out in space away from me. And you can attack those without getting really close to the middle element. And that's what carbon compounds do in their carbonyl manifestation. But that also makes them easy to hydrolyze, and there's a background always of amide hydrolysis. But you go to phosphorus and sulfur, and suddenly the sulfates, the polysulfates, the polyesters of phosphorus, the diesters, those guys can really be very hard to hydrolyze. And the reason is there's no pi bond available that leads you in towards the central atom. You have to go in at the atom yourself, and that's a huge entropy cost in stopping rotations and all kinds of things. So life really made a great play. She must have waited a long time to find that phosphate thing for the memory operation, but the fact that she picked it is really showing us that what we don't understand about it, I don't know why we lost the ball there. I would like to have Phil's ideas on that, because I just don't think the phosphorus and sulfur bonding are at all correct, and it's easy to understand why phosphate is so kinetically stable, and sulfates, when you make them. And that's what we found. With the QAC, it's very stable because we got a product that's like brick. The triazoles they're more stable thermally than benzene. They're really going nowhere, they're so stable. So, and then the sulfates and the phosphates, the sulfate diesters, they're stable up to 300 or 400 degrees if you have the right ones, and that's what we're having. Maybe I'll get a chance at the end or in the question session to tell you, we ran into another reaction that responded to the polymer version of click chemistry, which is polysulfates, a reaction that is blocking an electric field, the electromagnetic field, magnetic and electric. It's damaging. It comes through things from nowhere. It's kind of gets right through, and it gets into a capacitor, and it makes the capacitor get hot as hell, and also lose its ability to not leak its charge and devour itself. So that turned out to be totally accidental that we got into that. And so I'll try to talk a little bit about that later. It's really, to me, kind of shows the opportunity for basic curiosity-driven research to get into this strange idea of perfect reactions, and then to discover that one of them, the polysulfate one, is just as good as polyolefin polymerization, but it doesn't have any problem with water. Okay, so this is my favorite muse, the periodic table. So, and it got me into this perfect reaction, the QAC, which we had no right to discover, but just fell on our laps. And then comes the suffix, and it's just like it. It's almost unable to react with things that it's not ready to do. It's not programmed to do. So it's very strange. Okay, now, chemistry is, I got the idea over the years, and I'm not going to go into that. I'm going to write that down in a review article, but here you'll see that you have, the properties. I heard this talk at the ACS meeting, a physical Norris Symposium. For Hammett got the Norris Award for physical organic chemistry. And he said to all of us, the most fundamental and lasting objective of synthesis is not the production of new compounds, but the production of properties. God, that hit me right then. I should have known I was different, because I thought, that's exactly the way I feel. I don't like this. I was from a Norwegian fishing family, and I like to get fish in the box, and I didn't like any ideas about being elegant about doing it either. So I just wanted to get some function working. So that was fun for me to have that, and that stays with me. And nothing is more like click chemistry than that. And my wife, she's a writer, and she makes a hell of a mess of my writing. So the KISS principle, I'll never understand. I just don't know how to do it. And, that's a bad thing for any person that tries to write. So I don't write well. And keep it simple, stupid is what I remember, but keep it simple, sweetheart is a more affectionate way of saying it. Okay, chemistry's priorities. Well, Vince Lombardi, I think, said winning isn't everything, it's the only thing. And that's so cynical, but here's one. Reactivity isn't everything, it is the only thing. That came to me once, and I've never seen anybody could talk me out of that. Because if you're not doing a reaction, it means you're not having ability to dissipate energy and make bonds, and you're at equilibrium, and that's death. So we basically are constantly falling down across the gradient of energy, and that energy appears to be provided by the nuclear fusion in the sun, in our sun. So it's giving us the right to make stuff like human bodies and to erode whole mountains, and even to erode the earth. Now it's totally in charge. Life is totally in charge of the earth by having oxygen and acids coming out, lichens, and earth goes up and it comes down again with rain and acids. And so life is really powerful as a dissipator. And I don't think we see that as like a hurricane. My God, try to stop a hurricane. We get to see a lot about what makes them come back to life, but they are really powerful engines of creation of their physics-own organisms. things like that, and Bénard cells. And I think that's fascinating to me about the thermodynamics of chemistry and the condensed phase. The first creation was, in a way, it was the Big Bang happened. We're supposed to believe that now, and got a lot of protons and helium and some lithium, and that was like asking for it in a way. That was the most lowest entropy situation you could have, because now you're going to make suns eventually by gravity, and they're going to make they're going to have fusion and make heavier elements. And then we get the periodic table. So that's the first creation that we relate to. That first creation was the periodic table. And then we get the Earth, and we get eventually life somehow evolving on this Earth. And that has amazing, the reason why why the table could be made was apparent to a lot of smart physicists back 50 years ago, 60 years ago. They said, oh, well, the temperature is 200 million degrees Kelvin, and these things are happening way up there, and it's not, you can already see with our crude theories of physics that was what was happening. And then came black holes and lots of really vicious energy treatment. But down here on Earth, we got nothing but water. I mean, if we get out of 0 to 100 degrees, that's the magic range for water, we're violating life's basic assumption that she will hang on as long as there's some areas that don't go out of that liquid water range. And so it's so complicated, in my opinion, what life is. It's all self-referential, and we like to think we can see things and push buttons, but I'm not really counting on it for life itself. But still doesn't make you give up. We have things we can do, and I think engineering things that we need to do, or abilities, well, we might have to get off the planet. I don't take any credit for this, because I don't really see how we can get off the planet. We need a kit from biology to do that, I guess, but we're not going to get one of those. So people, the sun's going to eat us eventually, and that's fine. That's billions of years off, I guess, but it won't affect this beautiful town very much. It'll be somewhere else by then. Okay. That's too much time. Okay. Still going. Yeah. So dreams for click chemistry, where they use a few good reactions and make function. But we found out we couldn't do it. We couldn't even take the best of those reactions and get a compound in one go, join two big things together with acid chloride combining with an amine. It's not a perfect reaction. You get a mess if you do these things under general conditions. And so we didn't have a really perfect reaction, and we found that even all the other reactions except QAC and suffix would not deliver material for initial screen right off in one reaction of coupling. It's just not good enough, 99%. You just get a mess, or you get nothing, because in little increments of things that affect reactions, they add up on you, and you can't expect to get what you would get in ideal conditions. So that dream was really, that's why people failed in CombiChem too. They were making the wrong kind of molecules too. They were making too greasy molecules, but they also were slow as hell making these molecules, and they had to purify the things and get them out, even though they're doing CombiChem. That's not in the cards here. Not if you can do it with something that moves fast, and that's what we're trying to do. And by luck, we found these two perfect reactions. There's some more, like thiolene reaction and some Jaws found, but I think people will notice what perfect reactions are now. There's something which you can't stop often. I mean, you can do it in Minnesota soup or the copper QAC reaction in blood. It doesn't stop. I mean, it just goes, and it doesn't care about that it's in plasma of human blood, things like that. See, that's it. That's really alien to start off with. Okay. So, but the functions come from the connections, and that's what the reactions provide. So I make blocks that have nice connections, and then I can guarantee that I get one of these connections in one-off reaction in a well, and I can screen it right there. And that's what we needed, a perfect reaction to do that. Can you believe it? Most people are using imperfect reactions, and they're using excess up to the wazoo of stuff, and they're still having a hell of a time because it's really hard to get rid of excess of everything. You know, you need something that's quantitative right down on the ground, one equivalent of one equivalent, and you get quantitative yield. That's really, I don't want to sound too arrogant, but that's really what I'm trying to say. And the fact is that we got to have, I don't know, we were really lucky to get two perfect reactions since they're so rare. But I could show you other examples of it where if you have 99.9995, then you're going to be able to get a 500 mer without any trouble. Like in DNA chemistry, it's very important. Tom Brown and a lot of people, Thomas Carell in Munich, they ran 900 to 1,000 nucleotides that were all connected together. They ran down those and put triazoles without fail on all those acetylenes sticking out there with, and they didn't fail to get them all done. And that's pretty bizarre. So, okay, I'm not doing very well here. I'm sorry. I just tried it. Okay, where am I? This is the bigness of space, of chemical space, and the littleness of everything else. The best guess for the number of plausible compounds by Regina Bohacek, that's before this group with Jean-Louis Raymond, and also noting it was the, well, I can't remember the name of the man now, but he writes a column about chemistry, and he was highlighting how many compounds there would be. And if you had 10 milligrams of each possible compound, that was drug size, that you'd have more than the mass of the universe. And I remember asking Professor Peebles once, a physicist, astrophysicist from Princeton, actually at the place where Einstein was, at whatever, I can't remember the name right now, and I said, Professor Peebles, excuse me, we were standing out in the rain in Belgium, waiting to go into a church to have this award, an honorary degree. And I said, excuse me, Professor Peebles, can you tell me the mass of the universe? And he said, you mean the visible universe? I said, fine, that'll do. And so he told me, 10 to the 60th. And I said, what? 10 to the 60th, what? And of course, physicists, you know it, right? What they're going to call it, it's Daltons. Daltons, and it's not even honored with a proton, it's Daltons, 10 to the 60th Daltons. So that's basically the mass of the visible universe. Well, it goes to show you how much we can't understand based on sizes and powers of 10. Okay. Okay, okay, given that space is almost infinite of the chemical space, just simple stuff we can make, I suddenly realized that we better have some really good way to make the bonds, and that's more and more arguing for these two reactions, which have suddenly turned up to be perfect, I think. And so they enable a lot of things to be pushed against the wall, which you could never do without some sort of massive incompatibility of making anything else. Byproducts are everywhere. That's life itself. It makes lots of things, byproducts, and it doesn't care. I mean, it works. You know, life is really amazingly versatile and easy. If it's with us, like the immune system, it worked, and she gave up at some point. You can't wait forever for a perfect immune system. It's a miracle to have one, like we do, so we can walk around on Earth. And by the way, we got an aqua lung full of oxygen, the whole bloody atmosphere. So it's amazing to think about how much life has provided for us and for life on this planet. Okay. Well, I don't know if I'm making my point. Oh, thank you. I've got too many things I wrote down to remember what I was going to say. So, but actually, this is a duplicate. It looks, but it is cool that that Helmut Schwartz found that the usefulness of useless knowledge, and that most breakthroughs in research are not and could not have been planned. Rather, they appear like puck in entirely unexpected corners. That's perfect for our attitudes as scientists, thinking we know everything. And this is probably something I should pass on, but it's my favorite book by a non-scientist, well, by Kelly, Kevin Kelly. It's a 1996 book, so it seems old. The only things he's missing, he doesn't understand dissipation, his first and second laws, but he gets it. But guys, it's just a wonderful thing to see. A beehive, what could be a better image of embedded behavior and something we don't know where it is? You can study a bee with everything you can imagine, even things we don't know yet, and you'll never find a hive. But the hive and the bee are one thing. They are together. That's what makes it hard to understand why we expect to have a linear story. And humans really expect that linear story. That's probably our self-defeating arrogance. Okay. Oh, I don't seem to be able to get out of this. Oh, okay, so here we are again. Reactivity enables bonds to be made and broken, and so you can make connections. So you connect things that aren't connected. That's the biggest message chemists can make in our world, because that's a really difficult thing. That's entropically difficult. So you connect two things, and then are they different? The chances are they are very different. And so that's where the properties come in. It's easy to make modules go together and get different things. And we're hoping that if we just have a few perfect reactions, we can just do a couple of them to get the assembly together, and these will have functions. They can be glues, or they can be they can be for friction, to relieve friction, or they can be drugs, or they can be as shields against electric fields like that. Like I remember in the space series, the fantasy stories, that put the shields up, and how you stop an electric field from coming at you and blowing you up with a bigger field. Well, that's what's possible, though. It turns out that the electric fields are real. For apparently this polymer we have, it doesn't like to go through those fields, and it stops, and then it doesn't bleed when it passes the current. And then we got a really great breakthrough, which is just about the pure in nature energy, and it's machine learning applied to this polymer thing, which made us able to take the temperature up another 100 degrees for the shield material. And so I'm beginning to believe that well, all based on perfect reactions too. That's really very bizarre situation, and I hope you don't think I'm bragging just totally. It's because I couldn't see it. We couldn't see. We thought we had perfect reactions. We really didn't know if we ran the numbers out that 1,000 steps wouldn't give you some mess. I mean, your hopes for synthesis, but we didn't know that. And so we always assumed that we're getting close to perfect reactions, then we just let it go. But a perfect reaction is brutally important in discovery world, where I need the certainty of chance. If I can make something by just uniting two things together, and I can make so many things that way, that should be enough to cover a lot of medicinal chemistry. And then I can make a ton, because the reaction's perfect, so there's no process chemistry. I long love process chemistry, but it's obviously just a patch-up job for a terrible discovery situation. So if you don't, so what we need is process chemistry as discovery chemistry. I always told the people I consulted for at Lilly and Merck that I wish I was talking to process chemists instead of medicinal chemists, and they just laughed at me. Oh, we make what they tell us to make, and they're so proud of that. Make anything. Oh my God, that's the way we are, I'm afraid. Okay. Okay, maybe I just, something's wrong here. I'm going to try to go this way. This is good. The blue marble. That's so uplifting for us, and we didn't see that until the space shuttle, things went out towards the moon, and they took pictures going away. You know, I like here. The Earth is blue. It's a blue ocean, blue. So much is blue, and then there's the clouds that are white, and then we have green a little bit, hopefully a lot, but we have dry land and green. But you know, Earth can't be green until it's blue. The green thing is a bit of a funny fact to me. I really think water is what the planet has in spades and what made it so lucky so far. And then we had this great experience with water ourselves. We found that if we just took water out of the tap and stirred it with two liquids that were these liquids down here, olefin, beta-pinene, and diazoethyl carboxylate, they do an Ene reaction. So that red thing, which looks like the Exxon Valdez oil spill, it just grossed out physical chemists. And I talked to them about it. I said, can you explain this rate acceleration, which was like hundreds of times, and it was boiling at the interface even. And they said, no, they didn't want to explain something ugly like a macroscopic oil spill. And yet, if you do this, it gets almost perfect yield. Ooh, I did it again. Oh, thanks. And well, this is, to me, people who work on biology and love biology and life, and you really need to think about it, because I think some other man who's just a biologist, he came to the same conclusion in the journal. I think he said at Colorado or somewhere in the Midwest, and he said he thinks all life's reactions go on in interfaces. There's a lot of interfaces and a lot of phase changes that we're just beginning to appreciate in water. And Philip Ball is very interested in that too, I know, and I got to talk to you about that. Because I just, you can't do much in pure water. It's got a high dielectric constant. It's going to short out all your great ideas for making dipoles and extended charges of communicating. It's just going to short them out. So it's not the best place to do chemistry for us, especially organic chemistry. And this was a big day for me, because I came over to see MG's student, Chen Wang. Chen Wang is up there in the beginning of he's now at Florida State, and he has a place in China too. He's a genius. He really came on strong right from the start when he went on his own. But he had this thing he wanted to explain to me, and he said he got, this 96% yield for 60 reactions. He'd taken a virus particle, which MG was collaborating with a very wise old virus person. They made cow chickpea virus. It has 60 addressable things on it in a certain manifestation. Then you can put on 60 alkynes, terminal alkynes, which was perfect for the HUAC, which was what MG had developed a few years earlier, and the bioconjugation. And so you could see it come up like a square wave. It came right up to 60 groups went on. You could tell by the extinction coefficient of the dye you put on. And so I asked him, I said, geez, that's impossible, isn't it? And how can you have 60 reactions in a row give you 96% yield? And well, it turned out that, okay, that's the corrected. I tried to fix this before, I'm sorry. This is the yield, 99.993 for 60 reactions run one after the other serially, you get 96% yield. So that was the yield for that thing. And I said, that's something nobody's going to believe, I think. So you better prove that to MG, and I talk about this a lot. He had to prove it to us, and he kept trying. Everything he could do showed that it was definitely a 60 very high yield. So he got what he said he did. And then we come to okay. It's 90. Okay, if we have a reaction that we're doing, let's see, how many times are we going to do this one? It's going to be, okay, it's 100 steps, and we, it's, what's happening here is if you have a 99% yield, I'm trying to show you how bad a 99% yield is. So there's 99% yield, and you get one of the thing, and that's not a problem for us. When we do one reaction, we get 99% yield. We're happy as can be in my life. So we get one. Okay, that guy goes away with the noise, so you don't care. But if you go 100 times and you have 99% yield, you go a little differently. You have 100 reactions. By the time you're at 100, you're getting only 37% yield of the fidelity, where everybody has the group they're supposed to have on in a polymeric way. And then if you look at that one, and whoops, damn, what happened? Oh, okay, I'm going to give up on that, but if you look at it, you got 1% or 0.06% or something. You're not going to have anything of this thing that was contingent on 99 or 100 reactions going smoothly. Here's a good one I like. This is old. We published this click chemistry, the first one in Angewandte. It was QAC, and then MG came along with a beautiful bioconjugation that's still the main one used today. I think that's run hundreds of times a day around the world, that fin bioconjugation, which takes QAC to grab stuff in biology. And here's one they use for years, because those of you who do DNA chemistry will know this bromodioxyuridine was a popular way. You'd make antibodies which would detect things that it had in it, but you could feed it to an animal or an organism, and it goes in, and you get, in places where uridine, you get the bromodioxyuridine, and then you go and you make an antibody to the double-stranded DNA. You go and see if you can grab it with oops. No. Oh, shucks. Yeah, okay. Oh, no. Okay, I have to go back. Oh. Is that going to do it? Try. I don't know which one to use here. Okay. Okay, let's do this now. This should have a sound coming with it if it's right. Maybe you can show that series for me. And so the antibody doesn't know what's there, and it really has to have what the old. It has to be carved up, and so it's carved up all right. And they put in acid and nitric acid. They just eat the DNA, and they come out with a wounded DNA, and now they can get their thing to bind. And no wonder the pictures with that old method of what you get when you get, they're ugly and they're sort of smeary looking. So two geniuses, in my opinion, Adrian Salick and Tim Mitchison at Harvard Medical School, they came up to use MG's bioconjugation to grab an acetylene that went into to actually got into the structure of the DNA by being pulsed in while the organism was dividing. And every time you divide, you make a new string of DNA, and they get some acetylenes in. And so then, here goes. So, whoops. Doesn't seem to. Okay, so now we got deoxyuridine in there. Now we got these little lit up ones aren't lit up yet. They're dark because they're coumarins, and the azide extinguishes the fluorescence or the phosphorescence, whichever it is, and so you have to wait till it makes the triazole by going into QAC reaction now, and the sound's gone. Oh well, it goes click, 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 and you get the QAC reaction. Oh, okay, that's it. And so now you've got these acetylenes in here, and you can grab them with QAC and make, well, where is it going to be? And then they published this as a method for labeling just fresh cells. You know they're fresh because the label is actually showing that they'd replicated their DNA. And there's one strand of new and one strand of old, and you can make them light up. And so what happens then is, okay, now this is a picture of confocal microscope where the cells are the woman who's long ago died, but her HeLa cells. And you'll see they're pretty big, and they replicate every less than 20 hours or less than a day anyway. And so they capture a lot of you pulse it in, ethynyluridine, and it goes in, and it goes to the nucleus, and it lights it up. And what we do now is make some holes with a little bit of damaging material to the cells, but they're not beat up totally. They just let stuff in. You put copper in, you put the dye in that will light up when it makes that. So that was, I saw that and I thought, my God, no wonder they're selling so many of these. I found out from the company they're selling like, at some points they were 100 a week or something, because biochemists love to see replication. Who wouldn't like to see? You can feed this to jellyfish out there, and they're not making all their cells all the time. You can see where they're making cells that day. Some little light up here and over here and inside there. And so and you can see notochords grow out in organisms like zebrafish. If you pulse it in EDU, they get, the thing lights up going away from you, and it keeps showing where it's growing, and basically it's such a cool thing that no wonder it's like more. Now, of course, nobody cites anything, and that's fine because it's just a routine thing they use all the time to get sort of track of their various cells and things they're trying to do. So that's an example of you can't possibly. I don't think, I mean, maybe I could have imagined somebody instructed me what I was supposed to be impressed by, but you know, you really can't imagine what things are useful for a lot of times. That's our problem. We don't know what the best thing to do with a lot of our chemistry is. And I know, well, Bill and I were talking last night or night before about his great protein chemistry. What can we do with it that's really important? And what can you do with it? And so you're always looking for something useful. And you know, screens are basically, we need a screeniversity, a university for screens. We've got a lot of fields, and they fight over their existence, and some of them are more important than others, but we don't have a screeniversity. I mean, can you imagine trying to come up with a screen for something like this electric thing that we stumbled on? I just can't. And, you know, I think that's our biggest problem. We've got these disciplines that need to have satisfaction in so many ways that aren't helpful. And then they kind of, people are proud when they put their lab coat on, you know, and it's, I don't want to belittle anybody who loves science and really wants to make a contribution. But I think one of the fellows who strong inference. What's his name? It was 1964, he published this article, and he said, unfortunately, everyone thinks that science is making bricks for the temple of science, for the future temple of science. But I'm afraid that most of those bricks are still lying around the brickyard. And worse than that, they're covering up what you need to see. This is really cynical, I'm sorry, but I think it's important to be cynical about things that matter to the whole world. We need to discover stuff that's real and put it on the ground and make drugs quickly. So that's what I really wanted to tell you. And I don't have the time to go on and tell you about the electric things. And I'll just want to keep Bengt happy and stop. I know I'm over time. But, and the other thing that's cool is that the sulfonyl fluoride group and on drugs and anything that's in the body, it's a sleeping beauty. It doesn't react if you're dead or if the proteins are dead. They don't react with anything covalently, but if they're alive, each protein, I think, could be having a single sleeping beauty, and you can annotate proteins that way. So they grab, here's an example. See, it grabbed, this one is an enzyme I don't remember. There's hundreds of enzymes that have been captured this way, and we've been involved in some of them, but I lost track of it. Here's the fluorosulfate group having reacted with a tyrosine. It loves tyrosines, it loves serines, and it makes this nice little capture, and then it went around and spun around in a circle and came back and crystallized in a form they'd never seen the protein before. So that upset them, that they don't, but the enzyme is wiped out. It doesn't work anymore. Okay, so I can probably leave some of these slides with somebody if they want to deconvolute them, but I had terrible trouble. I'm 84 almost, and I can't figure out all these little widgets that go. The Mac is so good.
Tell us what click chemistry is.
Oh, what a friend. See, he's a real friend, Well, click chemistry is reactions, the ones we found out. The best click chemistry is a perfect reaction, and there are only 2 of them. So that's upsetting, but it's your job to find more of these reactions. Because if you don't have a perfect reaction, you can't do anything in this world. You're going to go so slow over the ground of what you need to go fast over. Because the certainty of chance is that if you can go fast making things with function, you'll find them, and they will be the cheapest way, because you can make them quantitatively from the reaction you discovered them with. There's no question about this to me, and I would not expect to live to see this happen, because people have so many investments, and they put their lab coat on, and they really feel, this is something. It's not like that in science. You have to be tough on yourself. And those of us who are scientists that have done something, we really realize that maybe we are tough on ourselves, tougher than most people think, because it's so easy to go off and do stuff which you can see. Van Temelen told me, you should never do anything you can do. That's good advice right there. I mean, that's why we need to collaborate. Chemistry is simple. It's a connective game, and I've tried to reduce it to a simple connective game. We've got to make the connections to get paid for them and entertain people and get the probability of usefulness. If we don't make those connections, it gets harder and harder to support a science. But we need physics. We need biology. We need everybody's we need to go into those disciplines and do stuff there, because once we're trained in chemistry, we're pretty powerful, I think, and we can do a lot for people. But we can't stay in chemistry. We don't need to collaborate with other chemists. That's ridiculous. You got to go out into the disciplines that are currently exciting and have things they need. And we're the only ones who can make new molecules. And whatever you say about that, you're not going anywhere interesting unless you make bonds and break bonds. So that's where we live, and we can do it, and we need to get better at it. And I think we're there. I think we're better. So I suddenly feel very much better, because, it's just a miracle to see a quantitative yield in a messy system like human blood. You know, just serum. Can you imagine running your reaction in serum? Well, it works. Yeah, okay, thanks, Bengt. Yeah, I would love it, yeah.